All right, so good afternoon, everyone. First of all, I'd like to thank Dr. Nirula and the organizers for inviting me. It's certainly a pleasure. This is the first conference that I'm attending back after finishing graduation over here. So I've attended multiple conferences in the US and Europe, but this is the very first time I'm coming back home. So this is fantastic. So as Dr. Nirula mentioned, I'll be talking more about gene therapy. So this will be quite a change from everything that we have talked about so far. We've talked about CAR T cells, which are essentially T cells that have been manipulated. But what I would be discussing is hematopoietic stem cells, which are completely different. And so you have to completely forget what you heard so far and start thinking about HSCs from a very different perspective. Also, there are a number of clinical trials which are going on in this field right now and a lot of emerging data. So rather than focusing on the data and showing you some of the numbers of the 100-something patients that have been treated so far, I'll try and focus on some of the larger concepts which I think are important for discussion in this forum. All right, so these are my disclosures. I do want to state in the beginning that I do consult for, and I'm on the advisory board for a number of companies which are developing genetic therapies for hemoglobinopathies using CRISPR-Cas9 base editing, etc. But as I mentioned, I'm not presenting any data today, so these disclosures don't mean so much. I will be using gene therapy to encompass gene editing in this talk. I should tell you that some people who are gene editors, they really dislike when we call it gene therapy. So it's an emerging field and the nomenclature is improving and changing every day. I will be discussing the pathophysiology of sickle cell disease because not many of you might be familiar with that. And then we'll talk about the current landscape of the different genetic therapies. And then lastly, talk about the challenges and opportunities for development of these genetic therapies. And so very briefly, sickle cell disease, it's caused due to a point mutation in the hemoglobin, the beta globin gene. Now, hemoglobin is a tetramer. It comprises of 2 alpha and 2 beta genes. And the point mutation happens here in the 6th codon of the HBV, which is the adult beta globin gene. Now, there are other globin-like genes like the gamma globin and the epsilon globin, which are produced during earlier phases of human ontogeny. So when we are all in our mother's womb, we all make fetal hemoglobin. And around the time of birth, something happens and fetal hemoglobin gets turned off and adult hemoglobin turns on, and that's where the mutation is. So all individuals who present with sickle cell disease, their symptoms begin sometime in the first year of life as the switch is happening. And so the pathophysiology is quite simple. Because of that point mutation, instead of glutamic acid at the 6th position, we have a valine. And what this does is, normally your hemoglobin molecule is supposed to be tetramer, 4 molecules combined together, 2 alpha, 2 beta, and they should be independent from each other, giving rise to that biconcave circular shape of the red blood cells. But when you have valine at that position, and you lose oxygen in the deoxygenated state, these molecules, they join together to form polymers. And these polymers, they give rise to the sickle shape, the C shape of the red blood cells. And these red blood cells are hard, they are stiff, they get stuck in blood vessels, which gives rise to all the pathophysiology of sickle cell disease. So these blood vessels, these red blood cells, when they get stuck in the lungs, you have acute chest syndrome. People can't breathe because of that. When they get stuck in the brain, you have stroke. If they get stuck in the bones and muscles, these individuals have lifelong chronic and acute pain. So really a devastating disease for individuals that have this problem. What is also important is that this is a progressive multi-organ failure disease, something that we don't always talk about. And so many of these problems that we see with sickle cell disease begin in early childhood as the switching happens. And so little kids can have strokes, splenic sequestration, but as they get older, they start having more and more problems, kidney damage, bone damage, acute pain happens continuously throughout their lifespan, cardiomyopathy, etc. And all this leads to early death in these individuals. What is important to note over there is that many of these changes that happen in these individuals are irreversible. And so once a stroke happens, it's a stroke for life. You can't really reverse that. And this will be important when we start talking about the C word, the cure, by using gene therapies later on. And so this figure over here, it shows the mortality trend in the United States over the last 3 decades. So the black line is looking at mortality trend in the cohort accrued in 1970s, the green line in 1990s, and the red line in 2017. And so you see on the x-axis, there are different age groups. And majority of individuals, they end up dying when they are in their 35 to 45 sort of age group. There used to be a little peak in the early childhood around 5 years of age because of newborn screening and because of pneumococcal prophylaxis. That peak has now gone in the subsequent decades. But despite everything that we have done so far, despite all the advances in medical sciences over the last 30 years, we still have the median age of death in these individuals at around 42, which is about 40 years less than the median age of death in other individuals in the United States. This is my favorite slide to show over here. This is a cartogram where the shapes and the sizes of the different countries have been inflated to show the number of individuals with sickle cell disease that are there in these countries. And I don't think I need to point out the two main places over here. There's Africa and there's Southeast Asia, including India, which are really have the biggest burden of sickle cell disease in the world. The place where I practice in the United States, even though we think that sickle cell disease is a big problem over there, and it is, there are about 100,000 individuals with sickle cell disease. You can see that the United States is like a little sliver of scrunch over here. So the biggest problem is in sub-Saharan Africa and India, and so that's why I'm here talking to you all. These are the currently available therapies and the years in which they were FDA approved. So we've known about blood transfusions for a very long time. It's a blood disorder. We give them blood and they get better. Hydroxyurea was approved for adults in 1998 and then children in 2017. In the last 5 years, we have a number of other novel molecules which have come into the market as well. But none of these medicines is a cure. They all have to be taken on a daily or a weekly or a monthly basis. And they don't work for everybody. They work for some individuals. They don't work for all. We've also done, actually my institution, St. Jude, did the first transplant for an individual with sickle cell disease in 1983. So we've known about 30 years that you can use an allogeneic bone marrow transplant to treat individuals with sickle cell, but not everybody has a donor. And as you know, transplant has its own side effects, especially the immune side effects. And so it is also not the preferred therapy for many individuals with sickle cell disease. And as Stephen was mentioning earlier, hopefully the first gene therapy using a lentiglobin, the lentiviral gene therapy, should be approved very soon. And then the CRISPR therapy with Vertex should also be approved, hopefully in 2023. So we'll at least have 2 autologous genetic therapies for sickle cell disease within the next 1 year. I should mention over here that here in India, these are the only things which are available. So hydroxyurea is the only main drug which is still available that is used extensively for majority of the patients. So, so many of these drugs, even though they are approved, are not easily accessible and available, and they're extremely expensive. And so even though gene therapy might be a very expensive alternative, when you look at the lifetime expenses for using any of these drugs, gene therapy might still be cost effective. All right, so there are a number of problems with transplantation, as I mentioned, and the biggest problem is that not everybody has an allogenic donor. Those who have donors, they suffer from graft versus host disease, graft rejection, hair loss, which is usually temporary but quite distressing to a number of patients, infertility, risk of infection, and then a small but non-zero risk of either developing a secondary malignancy or dying during the process of undergoing transplantation. So how do we improve on all these is the big question, and that is why genetic therapies for sickle cell disease have been developed over the last many years. So very briefly, how do you do an autologous gene therapy? Let's say if this is a patient with sickle cell disease, and on the other side you can see what their blood looks like. There are a number of sickle cells over there. The very first thing that you do for this patient is put them in the hospital. Here, I put them in the hospital. And then you collect hematopoietic stem cells from them. Now, I'm not going to go into a lot of detail of how we collect hematopoietic stem cells. Previously, we used to do bone marrow harvest. That had some problems with it, but now we do peripheral mobilization and collect them from the peripheral blood. So we collect those cells, we send them to the laboratory, we grow them over there, and then we do whatever magic we need to do. So sometimes we do lentiviral gene addition, sometimes we do CRISPR-Cas9 to disrupt some regulatory elements. And then once these cells have been genetically modified, they are frozen and QA/QC ensues, while the patient receives conditioning regimen, which is mostly busulfan-based conditioning regimen in almost all the clinical trials right now. What busulfan does is it wipes out their bone marrow from all the sickle-producing cells, and now they have an empty marrow, which we can infuse these genetically modified cells in. We give them a couple of weeks. As these cells start growing, these cells start making the red blood cells, which are coming from the genetically modified HSCs, and the patient does not have sickle cell disease anymore. So this is, in short, how the process works. Now, I've showed it in here in 30 seconds. It takes about a year to get it from beginning to end. But this is what the process looks like. And so many advantages over here. First of all, you don't need a donor for autologous gene therapy. Every patient can serve as their own donor. There are no immunological complications because you're receiving autologous cells. Having said that, some of these other complications like hair loss, infertility, risk of infection, secondary malignancy, and risk of death still remains a possibility even with autologous therapies. All right, so this is the landscape, and now I agree that this is not as busy as the last slide Stephen showed, but nevertheless, this is a very, very busy landscape of the different companies which are currently developing different genetic therapies. So very briefly going through the list, on the extreme, that's your right side, on the extreme right side, you see lentiviral gene addition. So lentiviral gene addition is basically adding a new gene, a new normal copy of the gene to the genome. When people ask me, what does it look like? So you have a car which has 4 tires in it, and let's say one of the tires has a puncture. You add a 5th tire somewhere in that car. That's what lentiviral gene addition looks like. All right. So there are a number of people doing that, and certainly Bluebird has been the first. They've treated about 60, 70 patients, and needless to say, most of those patients are doing fine. Then you have fetal hemoglobin induction technique. So you can induce fetal hemoglobin, which is the fetal equivalent of the adult hemoglobin. You can induce that by either targeting the BCL11A enhancer, and a number of companies, including Vertex, which is the leading clinical trial doing that right now. You can also disrupt the regulatory elements at the globin locus where the switching happens. And that's the approach that we have taken along with Editas, Novartis, and Beam. And then the last thing, which is sort of makes sense, but is the most difficult to do, is to actually correct the genetic mutation. I told you it's a spelling mistake in the HBB gene. People would ask, why don't you just go and correct that spelling mutation over there? It's easier said than done. That's the most difficult thing, and that's why it's on the other side over here. And there are a number of people who are trying this approach. And like I was mentioning earlier, these 2 products hopefully will receive FDA approval within the next few months. And so what are the challenges to achieving a cure for patients with sickle cell disease? The very first challenge is I always tell my patients that their body is like a wall and sickle cell disease is like a hammer. The hammer keeps chipping on their wall every single day, and transplant or gene therapy can take the hammer away, but it cannot repair the wall. So as I mentioned to you earlier, there are a number of damages that sickle cell disease does to your body which are irreversible. And so all these patients who have enrolled on the Vertex and the Bluebird bio trials, after they've received genetic therapy, they have not required any further sickle cell directed therapies. They don't need hydroxyurea. They have not had any further sickle cell related events. They don't have pain crisis. They don't have any more strokes. But the damage that's been done to them already, the damage that their bodies have accrued, that is irreversible. So if they've had a stroke previously, they've had a stroke for their life. If they had cardiomyopathy, that remains for their entire life. So one of the things which I think is very important for us to talk about is how do we frame what a cure is for these patients, because some of these things might not change, and many of these patients will actually continue to require therapies for management of these complications, whether osteonecrosis or cardiomyopathy or stroke. They are going to require treatment for these issues lifelong, even after receiving a potentially curative therapy. So the key here is to actually perform these curative therapies before these irreversible damage accrues and happens to these patients. Of course, now that would require doing these therapies to younger and younger children, which remains a problem while the long-term safety signal of these therapies is not fully established. All right. What are the other challenges to any hematopoietic stem cell gene therapy? As was mentioned earlier, hematopoietic stem cells, they're way more complicated to collect hematopoietic stem cells. I actually envy all my CAR T cell colleagues because it's so easy. They go in and within a couple of hours, they apheresis, they've collected plenty of T cells. They can manufacture 2 or 3 CAR T cell products. To collect hematopoietic stem cells using apheresis, we cannot use GCSF in these patients because GCSF causes extreme neutrophil activation, and some patients have even died when we've used GCSF in patients with sickle cell disease. So the only drug that we can use is plerixafor, which is not very effective. And so it takes almost, we can do 3 to 4 days of apheresis at a time. And sometimes we have done 3-4 months, multiple episodes of apheresis to collect the requisite number of cells to manufacture gene therapy. The next is, of course, the off-target effect. So the Cas9 therapy that is more extensively used right now, and of course, lentiviral gene addition, those things have been more thoroughly discussed and explored as far as off-target effects are concerned. But with some of the more novel therapies like base editing and prime editing, we don't even have the tools to examine how are we going to check for off-target effects using these therapies. The next is pre-gene therapy conditioning and toxicity. So as I mentioned, busulfan is the standard prior conditioning agents that we use for almost all the therapies right now, and that is associated with a lot of conditioning-related toxicity. Safety, I'll talk about that in a little bit. And then ethical concerns, financial considerations, and equitable access in the regions where these therapies are mostly required. I mean, these are, you could talk about all these topics for an hour, and still you wouldn't be able to scratch the surface of these issues. So let's talk about some of the operational logistical challenges. So most of these challenges are currently associated with ex vivo hematopoietic cell therapy. Apheresis, as I mentioned, it requires multiple cycles to collect adequate cells. We are developing better mobilization agents which can be combined with plerixafor. One such agent is GrowBeta, which was being developed by a company called Magenta Therapeutics. So hopefully, with the development of these novel mobilization agents, we will be able to reduce the number of cycles these patients have to undergo mobilization to collect adequate cells. Of course, there is that need for myeloablative chemotherapy, which has both short and long-term toxicity. And so there are a number of companies which are developing non-chemotherapy antibody drug conjugates, which can be used for conditioning to deplete the hematopoietic stem cells. Hopefully, they will be coming into the market in the next couple of years. And I think they will be a very big game changer in the field of autologous gene therapy for different non-malignant disorders. And then, of course, in the next 10 to 15 years, we hope that in vivo gene therapy, where we don't have to take the cells out, modify, and then put them back, we can just put the vector or the nanoparticle, which is carrying the modification molecule, we can directly carry that into the body, and we can edit these cells within the person's body. The other question, as I mentioned earlier, is of safety. There is the historical context when we started doing gene therapy for HSCs, for 𝐁𝐤𝐤𝐸 and other immune deficiencies back in the 1980s. We were using gamma retroviral vectors at that time. And of course, as you probably remember, many patients who received those gamma retroviral vectors then had leukemic transformation almost 10 to 15 years afterwards. So that led to a huge concern, rightly so, about the safety of these novel therapies. I can tell you that several hundred patients have now been treated using lentiviral therapies, and not a single patient has had a lentiviral-induced leukemia. Well, Steve is shaking his head over there. I'll talk about that in a little bit. We think that CRISPR-based therapies are going to be even more safer, but that remains to be seen because the follow-up is even shorter for CRISPR-based therapies. But long story short, longer clinical follow-up is needed. So the reason we are kind of a little bit skeptical about the safety of lentiviral therapy, myself included, is because earlier last year, in 2021 February, Bluebird Bio actually announced that there were 2 patients that had developed leukemia who had participated in their clinical trial. Now, they did extensive follow-up evaluations after that, and there were 2 major papers, one in New England Journal of Medicine and another one in Blood Advances, that were published. And in these 2 trials, in these 2 studies, they basically took away the blame from lentiviral vector. They said that some of these patients, well, actually in one patient, there was no viral integration in the leukemia cell. So they said, well, if the virus were never integrated in the leukemia cell, it couldn't have caused leukemia. In the other patient, while there was viral integration in the leukemia cell, it was in a site which is not a proto-oncogene, which was not believed to cause any leukemogenesis. Long story short, they said that the lentiviral vector was not implicated in the development of leukemia. Having said that, though, I would argue that sickle cell disease is a leukemia predisposition syndrome. In fact, the very first patient that I talked to you about who received transplant at our institute in 1983, that young girl received a transplant because she had leukemia. She had AML and also sickle cell disease. So she received a bone marrow transplant to treat her AML. And when she recovered, we realized that she didn't have sickle cell disease either. So long story short, sickle cell disease is a leukemia predisposition syndrome because of the extreme turnover of cells that happens in their bone marrow. So they might be at risk of developing leukemia anyway. And then you go ahead and start integrating either lentiviral vectors, or you start messing with the genome using CRISPR-Cas9 or whatever other things that we are doing right now. Those in some way could predispose to leukemia. So what I want to say over here is that long-term follow-up for all of these therapies is extremely important, and regular follow-up for these patients is absolutely needed to establish their safety. We also want to talk about what are the patient perceptions about gene therapy. We did a focus group and a survey at our institution, and 63% of patients reported they had no knowledge of what gene therapy looks like. Half of them said that if there was any risk of cancer, that they would not participate in a clinical trial of a gene therapy. This is because these patients, even though they know that sickle cell disease is a bad disease, they're not going to die because of sickle cell disease tomorrow. They're going to die in sickle cell disease in the next 30, 40 years. So there is that fear of unknown that might happen if they take the leap, if they participate in a clinical trial of gene therapy, and then something bad happens to them next month or in the next 6 months. So there are these unknown immediate risks of gene therapy which create a problem. The other issue also is that genetic therapies are extremely difficult to understand. It's hard for us to talk as professionals and discuss what genetic therapies are and what do we do when we are doing genetic manipulation. It's extremely difficult to even then talk to a patient who sometimes have limited health literacy to what is happening to their cells, and it's an irreversible process which they're going to live for the rest of their life. So there are these difficulties associated with gene therapies. There's, of course, at least in the United States, where the predominant population who suffers from sickle cell disease is African American, there is a lot of mistrust of medical professionals and research in those individuals. Like I mentioned earlier, there is lack of information, and the messaging around these genetic therapies is problematic. It's hard to understand, the long-term risks are unknown, and so we do need a better informed consent process and better educational materials to talk to our patients about these genetic therapies. And one of the things which we have realized what we should absolutely do is we should engage our patients as active stakeholders when we are developing these therapies, so that we hear their voice and we understand what is that they need, what is that they like to have, so that we develop a therapy which is acceptable to them. Otherwise, we may end up developing, we may end up spending $100 million developing a therapy which then nobody wants. Next, we would go to the financial concerns and the issues related to that. It's one of the most expensive therapies. 𝐁𝐢𝐤𝐬𝐢𝐥𝐚𝐫, which is the bluebird bio lentiviral gene therapy, when it was approved in the European Union, I think almost 2 years ago, they wanted to charge $1.8 million. EMA said, no, thank you, we'll pay you $900,000. And Bluebird Bio basically withdrew from the European markets, saying that we will just focus on the American market. And in the American market, they released this product at $2.8 million, which I can tell you, it is a huge sum. And 90% of my patients who need a curative therapy for sickle cell disease will never be able to afford that. So I don't know how we are going to ever take this therapy to areas where it's absolutely required. So once again, I want to highlight that figure over there. No individual in Africa or India will ever be able to afford this therapy at $2.8 million a piece, which is just a crazy amount of sum being charged for these therapies, which brings us to the point that there is an opportunity for local pharmaceutical companies and academic institutions to participate in the development of these therapies locally in a decentralized fashion and to develop these here. I'm not going to cover the payment models. I think this is a complete different talk together. But what I did mention is that the local development mass production might be the way to overcome these. Equitable access is sort of related to that. Is the cure accessible to individuals who need it the most? So many low and middle income countries do not have capacity to deliver these therapies. Fortunately, India does, because you all are already doing transplantation and this is no difficult. But in many other countries, there are competing priorities. There's nutrition, there are other infectious diseases which the governments have to focus their efforts on. And so some of these therapies may not be immediately available in those countries. But at the same time, I would argue that there is an opportunity to leapfrog. People generally say that we first have to develop a capacity to develop transplantation and then we go to gene therapy. I would argue that if you don't have the capacity to develop transplantation, you don't worry about it. You can directly go to gene therapy because it's autologous. It doesn't require some of the monitoring that is required for allogeneic therapy. And so there are less logistical challenges. And you could do it much more simply, much more quicker in these countries without having to go through the whole hurdle, the problem of developing an allogeneic transplantation. And then lastly, are the regulators ready for that? So there are a number of preclinical endpoints which we can use, but are they sufficient to allow a clinical trial development? This again comes to the question of the potency assays, which I can discuss in the Q&A session later on. how much preclinical safety data do we need for a regulator to say, yes, this is a safe enough therapy, let's go ahead and do a clinical trial? I think this, again, requires a lot of academia, industry, regulator partnership, because, again, when we started doing this in the US, FDA did not really have guidance. They had developed guidance for CAR T cells, but they didn't have guidance for hematopoietic cell gene therapies. And so we had to work with them, and they've been very receptive. But again, this is an opportunity to work with the regulators here in India as well about what would be the pathway to introduce a clinical trial and then for a regulatory approval thereafter. And so to summarize, there's certainly hope that we have potentially curative therapies available for patients with sickle cell disease and other inherited disorders. The long-term safety and risks of these novel therapies need to be established. Overcoming the cost barrier, I think, would be the biggest issue which will allow us to upscale these therapies. We have to remember where the patients are who need these therapies the most and the global burden of disease, which is where the hype surrounding the cure comes in. Patient-centered and the culturally competent educational content is critical for dissemination of these therapies to these patients. And then lastly, I think we have to engage with our patients as partners from the very beginning to develop a meaningful cure for these therapies, because these therapies have the potential to change their lives in more ways than one. And we need to understand that. So with that, thank you for your attention. And I'll be happy to take any questions during the Q&A session, but you can also feel free to email me later. Thank you.